nice to be able to be at an in-person meeting, and also nice to be here with the Zoomers. Hi, Zoomers. And it's great to do it virtually and give people an opportunity to come here that otherwise wouldn't have. It's been a fun year. I think we've all learnt to adapt to different situations, and I give a shout out here to all the grad students. This was not the funnest time to be a grad student, and trying to get your research done and get to conferences when there wasn't a lot of opportunity to get out of your own house was a tricky time. So I'm going to be talking today about gene editing today and in the future. But any of you that have ever heard my talk know I have this dog slide that I pretty much always include in my talks. It's almost a good luck dog slide. And so today is no different about genome editing, which is maybe a topic that you've heard a little bit about. It's one of the newer biotechnologies. And the reason that I guess I get to speak about it is I've been working in the field of genetic engineering for many years. And some people are like, what's the difference between those two technologies? So genetic engineering, or GMOs, to use the more controversial term, is basically introducing a trait to a breeding program that brings a useful characteristic along. And I'll use the example, since we're in the middle of corn country, of, for example, BT, corn. So you bring in a gene from an insect, from a bacteria that makes those corn no longer susceptible to caterpillars. That's what genetic engineering was, and it's kind of an older technology, if you will. There was not a lot of precision as to where that gene integrated into the genome of corn. And what's different about genome editing, and it's appropriate we're talking at a precision technology meeting this morning, is that you can very precisely target to any location in the genome the introduction, perhaps, of a new gene, but also just tweaking the DNA within an animal or a corn plant. And it's that precision that's kind of new with genome editing, and which opens up opportunities to very precisely inactivate genes in the genome without necessarily introducing quote unquote transgenic or exogenous DNA from another species. And that's really the distinguishing feature of the two technologies, and where genetic engineering is kind of an older technology. It's been around for, what, 30 years with very safe use of over millions. I think there's something like 17 million farmers growing it globally. Genome editing is really kind of a new boy on the scene. And there are a couple of gene edited plants that have been commercialized. But if we look at the history of genetic engineering in animals, we've had a really hard time commercializing those products. And I'm thinking here of the one that's really been approved in the United States is the fast-growing Aqua Advantage salmon, which has finally received approval and is going to be, I think, sold for the first time commercially in the United States this year, despite the fact that it was originally produced as an event back in, I think, 1989. So more than 30 years to get to market. Very different trajectory to what happened with genetically engineered plants. So here we have genome editing, and do we have a potential to have a different trajectory for genome editing than we had for genetic engineering in animals, which really hasn't been available to animal breeders over the last 30 years, despite the fact we've had the technology. And that's really where the opportunity lies, I think, to have access to that. Okay, all right, well, let me tell you what the second slide did. It's kind of a hard topic to do without slides because it's a little bit complicated, but anyway, we'll see how we go. But I guess I will talk a little bit about some of the fears I have around genome editing is that, and I will get to this in my talk, but there was a comment yesterday about the F&DA, I think was how it was phrased, who is actually in charge of the regulation of genome editing in animals, and have been in charge. So this is my very famous dog slide, but you might notice that it's had a COVID update since the COVID. And so if you look very, very carefully, of course, breeders can make dramatic changes to animals based on selections for particular phenotypes. And so, of course, we can have a poodle that looks like that lady, but I'd like you to take a very close look at that Irish wolfhound in the bottom left corner and see if you can identify who might be the individual in this room that the Irish wolfhound is mimicking. And so I think we've all had a little bit of fun with Dare Bullock's hair at this conference. That's been kind of fun to watch people's reaction, but that was Dare before he got the vaccine and shaved his beard. So I thought he had a definite Irish wolfhound look going on there. So with any luck, I'm going to, with the next slide, play a little movie where David Bowie and I are going to do a little duet to celebrate the progress of animal breeders. And I'm an animal geneticist. I acknowledge my biases there. But I think this story is one that needs to be told because genetics is such a powerful thing.
Oh, yeah. Changes, turn and face the strain. Changes, don't wanna be a richer man. Changes, turn and face the strain. Changes, it's gonna have to be a different man. Time may change me, but I can't trace time. Changes, turn and face the strain. Changes, don't tell them to grow up out of bed. Changes, turn and face the strain. Changes, where's the shame you've left us up to our next event? Time may change me, you can't twist time. Strange fascination, fascinating me. Our changes are taking the pace I'm going through. Changes.
The growth of the poultry industry is kind of breathtaking. There was 79 billion animals slaughtered for food last year. And I guess I always wonder when I compare 1957 chickens shown on the top here against 2001 chickens shown in the bottom here, fed the same way, treated the same way, but just genetics. How many more birds would have been slaughtered for food if it hadn't been for that type of genetic improvement? And I think this is a really interesting look at the global cattle numbers. And so this little graphic shows 1961. You can see India there with about 175 million. And America's in pink and has stayed at around about 100 million or so over time. Went up a little bit in the early 70s, back down to about 100 million or so, such that today there's about 94 million versus about 180 million in India. And Brazil has really increased to over 200 million. But that's actually only half the story, and a story we don't see very often because of the way this is broken down. The next little graph I'm going to show you groups Africa as a continent. And there you can see India at the top and Africa second. America's still pink, and this is over time. Just keep your eye on Africa. It's second at the bottom, or second from the top right now. Keep your eye on Africa. I don't think we often think of Africa having 350 million cattle, and certainly, what's that, 3.5 times how many cattle there are in the United States. And I think that this is an important story because of basically what's in this slide. Look first at the pie graph. America's there, green, 6% of the world's cattle. You can see Africa is about 25% of the world's cattle. Brazil, 14, India, 13, Europe, 8, and then all the rest. And actually the remaining is a really big chunk there at 17. If you look at the colors, they match the colors on the bar graph to the left there. And so you can see Africa with 350 million in purple, and then Brazil. Map that against beef in gray, the bar that accompanies each colored and goes to the right bar. And what you see there is US circled in red, the #1 beef producer in the world at 18% of the world's beef with 6% of the world's cattle. That's why genetics is important, because what that does is it improves the sustainability of a kilogram of beef produced in the United States. That's why it's important to understand when global averages are used to discuss beef, that is not the situation that happens here. And if we look at this, this is greenhouse gas emissions from beef production expressed as a kilogram of CO2 equivalents per kilogram of protein. And what you see is the areas that have really big beef cattle populations, so India and Africa, have really high emissions per kilogram of beef. And I think this is actually where focus needs to be paid attention to, because improving the efficiencies of production in these areas is actually really crucial to trying to reduce the environmental footprint of cattle globally. So I think genetics is a really important part of this. And so let's get into the bovine genome. And it's ironic or amazing to me, it was only 2009 or so that we actually finished sequencing the bovine genome, all 3 billion base pairs that make up the genome. And of course, the genome is in every cell of the body, and it is broken down into the nucleotides that we see, the C's and T's and G's and A's that make the proteins. That's kind of the central dogma. And after we got the sequence back, we found out that humans and cattle are actually 83% identical. So if you think about it, you're kind of a genetically engineered cow, of course, because we have similar evolutionary origins. And since then, we did the first genome back in 2009. Since then, the rather ill-named 1,000 Bull Genome Consortium has sequenced over 2,700 bulls from all over the world. And what we find is this amazing genetic diversity amongst the different bulls, over 86 million genomic variations, and those are SNPs, single nucleotide polymorphisms, indels between the different breeds of cattle. And that is actually the engine that drives genetic improvement. That is what you're selecting on, is this genetic variation that spontaneously occurs and is the result of why different breeds exist and why different cattle are adapted to different environments. And typically, it's not true that a single gene can have a big impact on characteristic. Most characteristics are quantitative, but there are some where one gene can have a really big effect. And I give you the example of myostatin, the Belgian blue animal shown here, who has basically an inactivated gene called myostatin. And if there's no myostatin, you get myogo. And so you get that double muscle phenotype. And this is the type of dramatic change you can have just from one alteration in one gene. Not all traits are like that. Most traits are quantitative, but you can have a big effect on single gene traits by altering a single gene. With the sequencing of the bovine genome, of course, then along came genomics and the high density SNP chips. And I think we're all familiar in this room. And I kind of love this image. I took it actually at Neogen. And the chips don't actually look multicolored like this, but when you take their picture, they kind of flash up like that. And every little rectangle there is where you would put DNA, and you can get 50,000 genotypes back on those animals. And that got incorporated into our breeding programs. And I think at first, there was a little bit of an antagonism between the genomics and the quantitative geneticists, but that all came together. And we have this really nice system now where genomics actually just plays along nicely with the quantitative geneticists, and we get a more accurate EPD. And that's what we were after. And certainly thinking back over all the BIFs I've been at, and back in the bad old days about a decade ago, where we had like multiple different types of data coming in and everything wasn't integrated into a smooth genomic EPD. It was a confusing time. And I feel like that was the beginning. Now we have a much more integrated system, and perhaps that's kind of the stage we're at with genome editing. So let me get into the topic of genome editing. Basically, genome editing is introducing a double-stranded break in the DNA double helix at a targeted location in the genome that you tell the genome editor to cut out. They have fancy names, there's zinc fingers and talons and CRISPR, but really they're a pair of scissors. And they can introduce this double-stranded break with a pair of scissors. And what does the cell do when it's got a double-stranded break? It wants to stick those two ends back together. It doesn't like having its chromosomes cut in half. Of course, this happens a lot during recombination, and this is a naturally occurring process in the cell to try to stick them back together. If you go out in the sun without sun cream, you will get double-stranded breaks because of the sun, and your body will put it back together. So these naturally occurring repair pathways will try and stick it back together, and it can repair in one of two ways. It can go down the left-hand side there and basically stick it back together a little bit incorrectly, so you get a knockout, and that means that you'll inactivate a gene. Well, why would you want to inactivate a gene? Well, maybe that gene does something that you don't want. So maybe it makes pigs susceptible to PRRS, and you would like to inactivate that gene so that that respiratory virus can no longer infect pigs. That's an actual true life example of a knockout that has existed. Or you could try knocking in, where you go down what I call the right-hand side pathway there, where you provide some sort of a donor template that has sequence similarity to either side of the double-stranded break. And you can incorporate the blue DNA there. And that can be something as simple as maybe moving an allele from one breed, like maybe not growing horns from Angus into, for example, Holsteins. Or it could be, if you wanted to, introducing DNA from another species like BT corn. So it depends what that donor template is as to what the outcome will be. And when I say this to people, I'm sure cattle producers are sitting there going, well, how do you actually do that? How do you actually get editing reagents into the genome of a cow and make that change? And really, what we would like to do is introduce that variation into the next generation of our breeding stock. So we would like to, for example, introduce perhaps a characteristic like disease resistance into the next generation of our stock. And the way that you need to do that is you need to somehow get into the DNA of that animal and make that alteration. And I'm going to kind of go through the different approaches we have, because at the end of the day, what you want is this calf here. And he is a homozygous, non-mosaic edited animal that has the intended alteration in the homozygous condition. So how do I get that little purple calf with the 2 pluses? And there are a couple of different ways that you can go about doing it. One way is shown here, and this is basically the somatic cell nuclear transfer cloning route. And I'll describe what happens. In this case, I take an oocyte from an animal I don't care about because I'm going to remove its nucleus, and I am going to do my editing in a cell line, and that's represented in the pink thing in the middle there. I'm going to edit that pink nucleus, and I'm going to insert my intended alteration in the homozygous condition to that cell. And then I'm going to take the enucleated oocyte that I got from that donor, and I'm going to basically do a dolly cloning, a somatic cell nuclear transfer cloning that will make that embryo that will then get transferred to the surrogate a calf 9 months later. And the nice thing about cloning is that before you transfer that edited cell, you can confirm that you've got the edit you want. It basically, what you put into that cell is what you put into that cell. There's no guesswork. And you can confirm the genotype, and you can actually get pretty high rates of knock-ins, or the right-hand pathway, in dividing cells in cell culture. The disadvantages is cloning efficiencies can be low, and you are using a single cell line, so you don't have a lot of genetic variation. Not all cell lines clone well, and you're kind of editing the last generation, if you will. So wherever that cell line was derived for, it's not necessarily the next generation that you're editing there. So that's one approach. Another approach is that you can, oops, this is out of order, I'm sorry. You can take an embryo from a cow you do want, so you want the genetics from the female in this point, at this stage, and then you inseminate it, maybe IVF with sperm from a bull you do like, and then you introduce, in this case, the editing reagents to the 1-cell zygote, which is basically a developing embryo. Then you transfer that to the embryo, and then you will, and that's called microinjection or cytoplasmic injection. You can also actually introduce it with a thing called electroporation. And that is basically, rather than injecting it with a microinjector, you can basically zap it in there with electricity. And the advantages of using of doing it this way is that you don't have to clone the animal. In other words, you're going to be giving birth to an animal that hasn't gone through cloning, so you avoid large offspring syndrome. You can use a diversity of germplasm, and you're actually editing that next generation, and you can get pretty high rates of knockouts, or the left-hand side pathway. The disadvantage is mosaicism, where not every cell gets edited the same, and so you can have different cell populations in those animals, and that's a real problem if, for example, you don't get the edit in the germline, because as breeders, that's all we care about. We want that animal to pass on those edits. You can have relatively low numbers of edited born, and knock-ins tend to be less efficient in early embryos, so it's a little bit harder to do that right-hand pathway in edited embryos. And so basically, I'm just going to zip through that. Basically, the disadvantage then here is if you look at the calf, that cow surrogate dam gives birth, you can have those mosaic animals where you have some unedited cells, you have some maybe heterozygous cells, and you actually have to go through a breeding round in order to get to the homozygous edited knockout animal, which is over there on the left, which is our ultimate aim. So in the case of cattle, where you've got a long generation interval, that's a real disadvantage of this particular approach if you don't get what you want the first time. So that's kind of the two approaches you get. That's the mechanistics of it. But let's have a look at, well, how might we use this in cattle breeding programs? And so if we look at this table, this is kind of things that have been reduced to practice, as in someone's actually done this. Nothing's on the market at the moment. And also maybe a couple of examples where you could envision you might do something like this. So if we look just generally at the targeted trait or goal, we haven't gone off the track of, we still are interested in productive animals, healthy animals, and good product quality. So what useful genetic traits could we introduce to our breeding programs that would contribute to those breeding objectives? And so what I have here is different traits that have been targeted. So for example, you could target the myostatin gene and increase muscle yield. Maybe you can do it in a little bit more of a nuanced way, so you don't have some of the deleterious effects that are associated with double muscling, like calving difficulties. You could, for example, introduce no horns into dairy cattle. So the polled allele that we're all familiar with in, for example, many beef breeds could be introduced into dairy cattle genetics. You could introduce an allele for slick that's well known in centerpole animals. Maybe you could introduce that, for example, into animals that don't have that to cope with increased heat. Maybe you could alter the coat color of animals to help with heat tolerance. Maybe you could alter product quality by knocking out a milk allergen, maybe improve meat tenderness using some of the alleles that have been found at Mark, the kelpane and kelpostatin. You could increase omega-3 fatty acid content by actually introducing a gene that contributes to improved omega-3s. Disease resistance is always a biggie. And so I think perhaps one of the logical things that you might use this technology for, so you can see some examples here, eliminating the prion protein associated with mad cow disease, mastitis resistance, bovine respiratory disease, tuberculosis. And then those last two I'll get to at the end, because they're a little bit different. They actually kind of alter a breeding program design to some extent. But those first lot before so the ones I've just discussed, they're all single gene traits, or at least they're all single genes that affect those traits. And I think that is what we can do right now. But of course, many genes, especially ones associated with traits of importance to this audience, are multigenic. Many genes affect whether an animal marbles or grows to a certain rate. And so I think I see a little bit of a little caution right now because I feel like quantitative genetics and genomics just had got their act together and they're walking hand in hand. And now it's like, ooh, I wonder if we can use genome editing to, alter things. And so this is great for single gene traits, but it's not going to address multigenic traits, at least not yet. Maybe we'll get great and we can edit many, 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 many genes. But right now, I think we need to stay walking down the street with the First Lady there. So here's some examples of animals. And I guess I put these up just to show you, these are not Frankensteins, they're just animals. So here's an example from A&M where there's a myostatin knockout in the Lorraine, and the one on the right there has a knockout as compared, of course, to the naturally occurring mutation in Belgian blue that I showed you earlier. There's an example here from New Zealand where researchers basically altered a gene in black and white Holstein to make them, I don't know what color I want to call that, diluted brown and white. And they did that with the idea to adapt animals, Holsteins, to climate change. And actually they just yesterday came out with a paper showing that the genomes of those animals had the intended alteration, but they weren't altered anywhere else in the 3 billion base pairs that make up the bovine genome. And that's a really important thing because the FDA wants you to prove that you did not alter the DNA anywhere else other than your intended alteration before they allow these products to come to market. Here's an example of Chinese have introduced a gene that enables animals to be tuberculosis resistant, and that's a really big problem throughout the world. Of course, a zoonotic disease also spreading quite rapidly in the UK due to badgers, of all things. And I realize I'm a geneticist, but to me, genetic improvement, which is permanent and cumulative, and hopefully this audience agrees, is a better solution to disease than antibiotics and chemicals to treat sick animals. And that where I think is the sustainability, low-hanging fruit of application of this technology. Another New Zealand example here, removing beta lactoglobulin, which is a really important milk allergen. So this is milk, non-allergenic milk, if you will. And I will spend a little bit of time on this project, and I think you'll see a familiar face there in the audience. This was basically a project that was in collaboration with Recombinetics and Oryxalogen, who I believe are in the audience here. They introduced the allele from beef cattle that don't grow horns into dairy cattle genetics. And this was done back in 2016. And basically the difference between those two alleles is the recessive horned condition. There's a 10 base pair sequence that's replaced by a 212 base pair sequence that makes animals not grow horns. And of course, that's a dominant trait. Dominant trait. And so Recombinetics introduced homozygous dominant polled into these bull calves, which at Davis were then crossed with horned Herefords, so recessive. And so we got heterozygous, and guess what, guys? No horns. Are you shocked? I'm sure this audience would be just shocked to hear that a animal that's homozygous polled gave birth to a bunch of homozygous heterozygous animals that didn't grow horns. And so there were 6 animals that were born, and we've been following them and have followed them for 2 years. And the phenotype was continually not growing horns. Every day we phenotyped them, there just were no horns. It wasn't a very exciting thing to phenotype. After the first time you phenotype that, you're about done. And so what you can see here is a couple of the males on the left, along with controls that were from bulls that hadn't been edited, growing horns. And then the one female we had on the right there is Princess, number 1 there, alongside her control, who of course is Cinderella. And Princess was quite a famous girl, so there was a lot of interest with her from the media, and you can see her here. She was coming in for a glamour shot because she was going to be on the cover of a magazine, and so we cleaned her up a little bit so that she looked good for her glamour shot. And here she is, 15 minutes of fame getting photographed, and there's a photographer on the ground there. And I think 4 people came to take a picture of this cow. We had the photographer and their assistant and a director and their assistant. And there she is on the cover of Wired. And I was like, oh, wow, that's kind of a little bit of a sinister picture. I mean, that's what she actually looked like. And so I'm not sure why they felt the need to do that. And then I felt bad for Princess, but then I opened the magazine and I went, whoa, okay. I guess that's just what they do with this technology. So anyway, so how might gene editing be used to alter cattle breeding program design, and I'll just touch on a couple of applications to kind of make you think. So it's not only for altering single gene traits, but it could also be used, for example, a project in my lab where we use genome editing to copy the SRY gene, which is the gene that takes you down the male determination pathway, onto chromosome 17, an autosome. And so the idea here being that if you inherit the SRY, but you're also XX, which is normally female, that you will be phenotypically male. And so this particular bull, who's heterozygous for SRY on chromosome 17, shown here in yellow, should have basically normal 50% male offspring, then also 25% normal female offspring from the one that inherited the chromosome without the SRY. But we hypothesized that the SRY XX animal would be, in fact, a phenotypically male animal with hopefully the growth characteristics of a normal male, but would actually be infertile because there are other genes on the Y chromosome required for fertility. But in the case of a steer that's going to finishing, that's not a particular issue. And you could even envision putting the SRY on the X chromosome and getting 100% male offspring. So doing a little bit of sex sorting in the animal, and that I think is a real opportunity for not only beef being male, but also obviously dairy female, chickens female for the laying hens. There's a lot of different applications where we would like to skew the sex ratio to basically better suit the production system that those animals are destined for. And this idea is a really interesting one, and that is that what if you could replace the testicles of the average animal with the germline of the best animals of the breed. And this is what's called surrogate sire technology. And it's shown here on the left, there's a donor male. That's who I would like my genetics from. But I'm going to deliver it from that boar on the right there, who is going to have basically genome edited, inactivated his germline. So he's no longer able to produce sperm. And I'm going to transplant spermatogonial stem cells to that boar so that his semen will be derived from the boar on the left. And so when he's crossed with that female, the offspring will be pure blood. So how might that look on in the cattle example? And this is actually some work of Macy Mueller, who's a grad student here in the room. And you could, for example, envision you would like to have a tropical sire. Well, we know we can have a tropical sire and a tropical cow produce a tropical offspring, but maybe they're not as efficient at beef production, but they're well adapted to the environment. What you might like is a bull that's well adapted to the environment, but maybe carrying genetics from, for example, a temperate animal. And what that enables you to do is basically have a surrogate sire that's maybe delivering Angus genetics to a tropical cow to produce an F1, but it's all done through natural service. And I think I was asking Macy to help me with describing this in a way that would be easy to comprehend for the audience. And basically what we came up with was producing bulls that shoot other bull swimmers. That's what this project does. And so that is a really interesting idea to think about how that might be used to deliver AI to an artificial insemination through natural service to external, particularly range animals that are difficult to otherwise get natural artificial insemination delivered to. So to me, I see editing as a cherry on the top of the breeding sundae. It doesn't replace breed associations. It doesn't replace performance testing, progeny testing, artificial. It doesn't replace any of that. It's the cherry. And if you can have a cherry on top of your ice cream sundae, that's a good thing. You can still have a sundae without a cherry. If we can't use the technology, we'll still have breeding programs that are incredibly efficient. But this does offer a way to introduce useful alleles without linkage drag and potentially bring in useful genetic variation from other breeds. And I think that that's the opportunity. And how do I envision it might impact a breed association or future of genetic evaluation? It really offers an opportunity to repair deleterious conditions, introduce useful alleles for single gene traits into breed germplasm. Currently, that's where we're at with the Mendelian simple traits, and it could potentially alter the defining characteristics of a breed. And I kind of think of, what could I do to this particular breed? Well, I could remove its horns and make it slick. And, you could make all sorts of changes that maybe would no longer even look like, in this case, a highland animal. And so those are the types of potentials that we have available. But the question is, will we actually be able to use it? And the reason I put that caution out there is that January the 19th, 2017, so one day before before the Trump administration took office, the FDA came out with a guidance for industry that basically said that any alteration that's done using genome editing is going to be treated as a new animal drug using the same kind of approval process that's required for ivermectin, for example. And that includes insertions, substitutions, and deletions. And so that's basically, even the left-hand pathway is being treated as a new animal drug, even though you're not introducing any novel DNA. And it's exactly analogous to the very variation that you guys all select on because it's been introduced but with naturally, if you will. And so each specific alteration that's intentionally introduced is going to be treated as a drug. And despite the fact I just told you, there's 85 million variations that occur naturally that aren't regulated. And so what it says, if you want to introduce any food from these animals into the food or feed supply, you need to go through the FDA authorization to get the INAD, or Investigational New Animal Drug approval. And so we have this dichotomous situation now where plants are regulated by the USDA and they do not treat alterations that could have occurred naturally, i.e. the left-hand pathway, as any different to conventional breeding. That's why we've been able to already commercialize genome edited plants in the US. The FDA, on the other hand, said, did you use molecular techniques to intentionally alter the DNA? If you did, you're a drug and you're going down the multi-generational, multi-million-dollar drug pathway. And there is no exceptions. So that puts the US in a very disadvantageous situation competitively relative to some of our big competitors. So would a gene-edited knockout food animal be subject to additional regulations in this country? It's a bit of a crapshoot, but South America, no. Australia, no. Japan, no. Canada, most likely not, depends on what the product is, but we've got 3 sticking here. EU, full GMO, well, big surprise. New Zealand, they're just a little EU, no surprise there. United States, that's a surprise that they're going to treat this as a new animal drug. And I certainly didn't expect that decision to come out. And I think it's a very, very bad one. In the case of genome edited knock-in of an endogenous allele, so for example, polled, that would be subject additionally to regulation in Australia because they use a nucleic acid repair template. So just recently, just before Trump went out, and this really isn't a political issue, this is a science issue, and it's kind of frustrating that this is kind of getting a little bit caught up in politics. But the USDA has put in a notice of proposed rulemaking to transfer regulatory authority of genome editing in food animals that basically could have been achieved using conventional breeding to the USDA, and that would include cattle. And I see this as a positive thing because I don't think the FNDA approach is workable, and it hasn't been for 20 years. And so there was a Federal Register comment period that was closed May 31st, 2021, and they had 51,239 comments. If you want to raise your blood pressure, go read those comments. There's just a lot of interesting things in there, but this is really very much up in the air as to what's going to happen next. So I see so much potential for genomics and genome editing and reproductive technologies to form a troika in the future that can really power breeding programs. But it's unclear to me whether we're going to have the regulatory ability to bring these products to market. And so I thank you for inviting me to BIF, and I apologize for the technical difficulties and running a little bit long. Thank you to the funding sources that have been supportive of my work. And I'll just finish with an invitation to anybody that's interested to the World Congress on Genetics Applied to Livestock Production, which is in the Netherlands next year. So thank you very much, and I'm happy to take questions at this stage, I think is probably how this goes. Thank you. The question, I think, was whether there's any pleiotropic effects in the embryonic stage or post. It depends, of course, what the edit is. So if I edit an essential gene, there will be no post embryonic stage because they'll be dead. And so I think it depends very much what the edit is as to what the pleiotropic effects might be. There will, of course, when we're knocked out a gene that we haven't previously knocked out before, then we have to look at what potential pleiotropic effects there might be. And that's part of the as any breeding company would, you look at, is there other unintended, or I think we call them correlated traits, right? That have negative correlations with the trait of interest. That's language that we would use for that. The unintended effects sounds like something kind of really sinister, but we always look for negative genetic correlations with traits that are of interest to us. So I don't see that changes very much just because you're edited. What does concern me is the assumption by the regulatory language that there's going to be these really draconian alterations. And so you have to prove, for example, in the case of the horned animals or the polled animals, we had to do, for example, meat and milk composition analysis. Well, what's polled doesn't affect meat or milk composition. It wasn't a biologically hypothesis getting tested. It was just something you had to do to tick a box to get to market. And that, to me, if we go down that rat hole, you know, you can spend a lot of money showing that meat is meat and milk is milk. And so to me, let's focus on biologically relevant hypotheses. So if you see an abnormal or undesirable phenotype, follow up on that. But to spend a considerable amount of money doing analyses to show quote unquote substantial equivalence for a trait that's not expected to affect milk and meat composition just makes this so prohibitively expensive that it will absolutely shut down academic research in this field. And I would argue even small companies, and leave it to the purview of large companies that have the kind of resources that can afford to do that kind of evaluation.
Dr. Van Eenennaam, I want to say thank you for your presentation today. And 2 years ago in South Dakota, when you first presented, or my first exposure to gene editing, I was fascinated and have been enamored with that. And I want to say to everybody in the room today, as influencers in our industry of registered breeding cattle, I encourage you to develop policy to register and genetically identify gene-edited animals. And I want to compliment the Red Angus Association for taking a very proactive approach to that, to say, and they have approved the registration of 3 different gene edits already in Red Angus and have some of those registered. So I think all of our breed associations, we need to develop policy, because the opportunities ahead and what we can do with health traits and what we can do with probably methane production, carbon footprint, that kind of stuff, to me is amazing. So thank to you and to Macy and all of you that are doing work in this. Also, there is another session this afternoon in the breakout sessions that you may want to learn more about gene editing and how it affects us as breeders.